For Chesapeake Bay Research and Restoration, and our moderator for this panel is Larry Sanford from University of Maryland Center for Environmental Science. Larry.
Thank you, Raleigh. Hi, everybody. Again.
Good morning.
I'm really quite honored and humbled to be up here because I'm not an SAB researcher at all. But I'll show you in a minute my connections to SAB. We have some distinguished panelists here. We have the old garden, we have the new garden. Our panelists are Bill Dennison from UMCS, J.J. Orth from BIMS, Cassie Gurbisz from St. Mary's College of Maryland, and Jonathan Lefcheck from Bigelow Lab. J.J. and Bill are professors, have been for quite some time. Cassie is an assistant professor, and J.J. is a postdoc at Bigelow. John. Not John, I'm sorry. I got this. Would you go to the... So, me, why am I up here? I'm up here because I've been a benthic physical oceanographer for my entire career. And physical biological interactions are really, really key in the benthos. It's one of the only places in the aquatic ecosystem where biology really affects physics, and I find that really, really intriguing. And I got converted to Team SAB in 2010. We were doing some... Yes. We were doing some reconnaissance surveys for an Army Corps of Engineers funded study up in the Upper Bay, and we drove our little boat into the middle of the Susquehanna Flats without even realizing we were there, or at least that we were in the middle of the grass belt. And when we stopped, it was just amazing to me. The water clarity in that environment, which is the thing that I really care about the most, is water clarity and suspended solids, was just amazing. I thought to myself, if the whole bay could be like this again, it would just be wonderful. And also, because I worked for many, many years with my colleague, Eva Maria Koch, from UMCS, who died in 2014, and she was also completely devoted to SAB studies and to studies of physical biological interactions, and I feel very strongly about carrying that legacy forward. So this morning we have a series of interactive, they're all sitting up here because they're going to go back and forth and pass the baton and make it really interesting for us. So I'm going to sit down and let them just go. And at the end of that, at the end of the presentation, I'm going to stand up and run the question and answer session. When we do have the question and answer session, I'd ask two things so we can get this recorded better. One of them is if you're near the microphone and you can get there quickly, please go to the microphone. Or if you're just seated, speak really loudly so everybody here can hear you and so we can get you on the recording. Okay, thank you. Take it away.
All right, I'm going to kick things off here. We're good? Okay. Let's step back in time a little bit. We want to talk about the story of SAB from the beginning. And in Chesapeake Bay, it's been a prominent feature from the time of John Smith onward and prior to John Smith. And you can see two aerial photos of Solomon Island down in where our Chesapeake Biological Lab was established in 1925. We see 1933 the aerial photo, pre-satellite, with dense beds offshore. And then you can see the pier was constructed in 1938, and then the beds were still remaining in 1969. But along came Hurricane Agnes in 1972. And that really was an important moment for resetting Chesapeake Bay. It was the first hurricane in 72. It happened in mid-June, so it's very early for a hurricane. The grasses hadn't even gotten established. Tremendous human impact on loss of life and property. Still today, the highest recorded flow at Susquehanna, almost overtopping the dam. I like the curves. So this huge event was devastating human terms, but it also was also very relevant in scientific terms, because what happened, some federal money came available. That's President Nixon looking at the overflight of the damage. And so the three big, they call them the big three, the three leaders of the Chesapeake institutions at the time, Bill Hargis on the left, the BIMS director, Don Pritchard in the center, the director of the Chesapeake Bay Institute at Johns Hopkins, and then on the right, Gene Cronin, the director of the Chesapeake Biological Lab. They got together and obtained federal funds. Actually, they sent people out in the field with the promise that maybe they'd get funds, but they thought it was so important they started the monitoring straight away. There's a book that's been produced that documents the impact of Agnes on the bay. And after Agnes, a lot of the grasses were gone. Back to the Chesapeake Biological Lab, 1999 aerial photo, and everything since 1972. Grass is not there, and even with potential transplants at the time.
Well, you know, a lot of you weren't even born before 1972, but I was right in the middle of my dissertation, and I remember going out and in sites that I've been using for my dissertation totally disappeared in front of my eye. I remember going to my graduate committee going, what am I going to do? Well, obviously here I am, and I got my doctorate. But you know, between Agnes and the next decade, a major program was instituted in which many of us who started our career back then began studying SAB. And one of those was we documented the decline of SAB in the Chesapeake Bay. And we went back to historical records and noted that what we saw in 1972 had never been seen in the history, recorded history of the Chesapeake Bay. It wasn't just one species, it was all species throughout the Chesapeake Bay. And we documented it in this paper that we published in Science in 1983. And another seminal paper that came out after that, based on research by the University of Maryland people, based on a lot of work that was done in Cambridge with the ponds that you see there by Mike Kemp and Walt Boynton. Again, two people who have been involved in SAB research. You know, linked the decline primarily to nutrients and sediments. And while there was a lot of controversy about pesticides, that really didn't fall out as one of the important predictors. It was really nutrients and sediments that became the overwhelming issue trying to figure out how to get SAB back. All this work that we did led to this historic agreement in 1983 signed by many of the dignitaries, the governors and dignitaries of the 5 states that led to the creation of the Chesapeake Bay and the life of Rich Patuk began that we talked about in 1983. And many of you know Rich. As part of this program in 1984, a significant monitoring program began. Most of it was related to places within the bay and the tributaries. Those dots were sites set up for both monthly and annual monitoring for water quality. And of course, we had set up an annual monitoring program started in 1984, where we looked at SAB coverage every year on an annual basis through today. And I do want to at least at this point say thank you, Rich. We wouldn't be here today without your support.
So after about 10 years, we did our initial synthesis. We called this Technical Synthesis 1. It was where we started to relate habitat parameters to the survival of SAB. And we have a fairly simple model here on the right, the light attenuation due to stuff in the water column, phytoplankton, measured as chlorophyll A or photosuspended solids, with nutrients that influence the chlorophyll, and the epiphytes growing on the surface of the SAB leaves reduce the amount of light reaching the plants. We also came up with this concept that the depth limit was limited by light, and it was approximated in Chesapeake Bay the Secchi depth. When we went and looked at this on a broader scale as part of this, as a bioscience paper we published associated with this Texan one, we called it the canary in the coal mine, because what miners in coal mines used to do is take a canary down. The canary had higher sensitivity to carbon dioxide than humans. So the canary would fall over, and that gave humans time to go, oh, we better get out of here before we fall over as well. So we've replaced the canary with the seagrass. The reason is seagrasses need more light than other plants on the planet. They need 10 to 30% of incident light, whereas algae needs 1 or less percent. So they can grow to much deeper depths. They can grow in much more turbid, low light environments than seagrasses. So the seagrasses are the canary. The second synthesis, Texin 2, followed up in 2004, about 10 years later, and this was refining those habitat requirements and bringing in Eva Marie Koch's physical exposure as well as sediment characteristics and a little more sophisticated light model.
So, while Bill and I were very interested and concerned about what was happening in the Chesapeake Bay, obviously our heart was in seagrasses in general, and we were obviously aware of some of the changes that were occurring in seagrasses worldwide. So Bill and I put together a proposal we submitted to a program out in Santa Barbara supported by the National Science Foundation called the National Center for Ecological Analysis and Synthesis. And that proposal was funded, and we brought together a team of people from all over the world, and it set the template for where we were going to be 10 years from now, not knowing that, but we really felt that from that effort, we had a method for how we would understand trends in seagrass. And so we had both the young and the old embedded, and one of the first products that we put from that was a paper in Body
I
Science.
want to say one thing. Larry mentioned Eva Marie, who passed away in 2014. Susan Williams, one of our colleagues who actually got her PhD at Horn Point, passed away earlier this year in a tragic car accident. So, tribute to Eva and Susan here.
So again, Bill and I felt it was important in order to energize the people. We put together very quickly a paper that we published in Bioscience called The Global Crisis for Seagrass Ecosystems, and a well-cited publication right now in the literature. And in it we have a series of conceptual models that talk about the ecosystem services and drivers for seagrass cover both in tropical and temperate areas for all the different species of seagrass that occurs worldwide. We also put together a map of the seagrass diversity in the world, where you have hotspots in the Indo-Pacific. Again, another well-cited article. But the major point of this whole workshop was to look at the trends, and we published a paper in Proceedings of the National Academy of Sciences that talked about the trends in seagrass cover throughout the world based on all the studies that we gathered that go back as far as 1879, and published this little map where the majority of the changes in seagrass were negative. There were some positives and some neutrals, but the major changes that were occurring in seagrasses worldwide were declines, which again stimulated a lot of interest in worldwide coverage and what we can do to try to get it back. And from that, of course, came the development of the International Seagrass Workshop Group, which is meeting right now in Singapore. And I told my colleagues to please get Trump and Kim Jong-un to sign a little booklet.
It turns out that conference that runs every 2 years, we're going to host it in 2020. So we're bringing the seagrass nerds of the world to Chesapeake in 2 years. So one of the things that we also did as part of this NC's effort is we started to look at why people didn't care about seagrasses. I mean, they just didn't have any charisma. What we did is we did a study, we added up all the scientific reports on seagrasses, corals, salt marshes, and mangroves, and we went and looked at all the media reports of those associated ecosystems, those coastal ecosystems. And what we found, to our surprise, was that seagrasses are very underrepresented in the popular literature. In fact, we put a proposal to National Geographic to have a seagrass article, and there's seagrasses as backdrop to seahorses and sharks and stuff, but never seagrass on its own. So, I mean, I don't understand, because we are just dripping with charisma, JJ and me, but the media not so much. So, so that was a bit of a revelation. We recognized we needed a little bit more charisma for our favorite coastal ecosystem. So kind of in summary of our trajectories work group, we established that seagrasses are sentinels of ecological change. We established that they're widespread and accelerating. In fact, it turns out it's about a soccer field every half hour is lost globally since 1980. So it's a real problem. And we needed a global seagrass conservation effort. We need to reverse these trajectories. And as we're going to see in Chesapeake Bay, we think we've done that. We've tried a few little things. We've got a seagrass blue carbon blues video we put out there, and it's gone bacterial. There are 4,227 views so far, so someday we'll get to our viral dissemination. We initiated a third synthesis, and this one wasn't sponsored through the Bay program, but it was something JJ and I took upon ourselves, and we brought together folks here in Annapolis to look at discern what we could out of the water quality and seagrass relationships. We established that we really needed not to talk about SAB at a Bay-wide scale, but talk about it at these regional scales, because different trajectories were happening in different places due to different causal mechanisms. Water clarity in some places, nitrogen in other places were the dominant correlates. So that started us on this path that we're going to see come to fruition with the more recent studies. We also, as a result of the Monitoring Realignment Action Team, MRAT, proposed a Chesapeake Synthesis Center back in 2009. And we said, well, we've had estuarine science since 1925 in the Bay. And that's really the establishment of estuarine science in the world was here in Chesapeake Bay as well. The establishment of eutrophication science is very much a Chesapeake phenomenon in the 70s and into the 80s. And then this coordination phase of modeling and monitoring, where the Bay program and the BIMS studies started monitoring the Bay, and the first physical model and then ultimately computer models of the Bay. And then this phase where we're using TMDLs and report cards to track the progress of the Bay. And we thought this is the perfect time to start to bring scientists together. So this is what we wished for, and lo and behold, our wish came true.
So, every 5 or 6 years, I've had to apply for our annual monitoring program. In 2016, in that RFP that came from Rich, included a component called status and trends. And so obviously, we applied for the grant, and in it we put together a rather progressive approach to looking at SAB. And when we were doing the proposal, I called Bill and said, Bill, let's do what we did in NCs. Let's now take the approach of putting together a key synthesis team and approach looking at the status and trends of SAB in the Chesapeake Bay. So in that proposal, that's what we proposed. And so it got funded. And in that very first meeting, we met with Rich, and Rich said, great. But then, knowing Rich, and you all know Rich, he puts this list of 1,000 questions in front of you. He said, here is what I want you guys to answer. And we go, so what Rich does, he says, I want everything you ever know of SAB from biblical times. I want you to perform a miracle. And so we go, oh my gosh. So Bill and I's first expression, we were sitting there, we said, oh my gosh, how are we going to do this? It was like, ugh. So, we took that, we stepped back and said, okay, let's put together a team of people, both young and old, and we looked around the Bay community, and we, based on our experience with what happened at NCs, we identified people that we knew could contribute, be productive, brought in talents of young and old, and this is our synthesis team. And this is the meat of where we're going to be going with the talk from this point on.
So we distilled that wish list into 2 fundamental questions to pose to the group. What are the long-term SAB trends in Chesapeake Bay, and how are the trends related to human activities? We felt like if we answered these properly, we could actually work through the wish list and answer all the specific questions. But first, we had to get to the nut of it. What's causing the trajectories, and therefore we can start to manage them more effectively. So, and part of the rationale for the timing of this was that Now, at this point, our first census was after 10 years of data. Now we have 30 plus years. So now we have long-term solid data sets that we can really interrogate. We've had droughts and floods punctuating throughout that time period. We had access to new analytical tools and expertise, really smarter, younger people. We recognize that these drivers of these trends can have really important management implications and relevance. And at the time when we started this in 2016, the idea was to fast track this to provide input for the 2017 reassessment of the TMDF. So it's timely to insert into a management context. And JJ and I felt strongly that we also needed to transition to a new generation of scientists, and so we brought along some good young scientists. And we had very specific criteria for selecting our participants. We wanted excellent scientists. We wanted, like Raleigh's thing, no known jerks. Focus on analysis and interpretation, a commitment to Chesapeake Bay, and a willingness to work collaboratively towards a common cause. And I use that Aristotle's term, praxis, thoughtful, practical doing. So it's not good enough to be the brainiac and just be thoughtful. We need people to be practical that would help us get to these management implications. So we set up a series of workshops. We did them short burst of time, immersive workshops. They were productive. We produced a blog after each one summarizing what we did, bookmarking our event, documenting the process. They were very interactive. The activities and breakouts really led to input and exchange. Nobody sat in the corner working on their emails. They were condensed. We had 2-day, sort of two and a half day workshops, limited to the necessary contact hours, and they're participatory, multiple opportunities for input.
So in that very first meeting of our group, Bill and I reflected again on how we were successful with the NCs group back in 2006. So we said, okay, in order to generate the enthusiasm of this group, let's get a paper pretty quickly, just kind of talking about the general picture of SAB and what's happening. And so within the first month, we outlined and put together a paper, and I hate to say this, but I remember I was in Australia on part of another project in a room during election day, putting the final touches to this paper. And I told the people, don't tell me what happened with the election. But post-election, this is the paper that came out in Bioscience, SAB and Chesapeake Bay, Sentinel Species in a Changing World. And again, we wanted to generate the enthusiasm of the group because having stuff in the literature for a lot of us, especially the young people who are building their career, we wanted to make sure they had something to show for this effort, because we were asking them a lot. They weren't getting support except for the money that we had for a postdoc. But there are many other people that were essentially donating time, and we felt, let's do something that gives them something that expresses our appreciation for that. So here comes the paper, and with a cover that came from Cassie's work on Susquehanna Flats, and we were really stoked with the fact that we had a nice cover. And in this paper, we talked about a number of things, one of which was the fact that SABs are not just an indicator of water quality, which is, of course, one of the emphasis of this effort for the Chesapeake Bay, but also we call it a defender, for defender of water quality, defender of habitat, defender of shoreline. And this is a little diagram that we drew at a restaurant at a dinner by Chris Patrick, and which is how we sometimes worked. As Bill said, we never left the effort, whether it was drinking beer or a dinner. We're always talking about stuff in between all the fun things, and these are some of the things that came from that. And in this paper, we described some of the trends for the different sections of the bay, because the Bay SAB responds to different parameters, different parts of the bay, which is going to lead to the ultimate product that John will be talking about. In this paper, we want to show some of the trends that were occurring bay-wide and in different segments of the bay because they were different. In this paper, we put together a series of conceptual diagrams for case studies, which you'll now hear. But we wanted to point out that there were a lot of things going on in different parts of the bay that were different. So one of the key things that were changing in the bay that we wanted to highlight in the lower corner, right corner, was aquaculture. And some of you, if you are familiar with looking at Google Earth, aquaculture or keeping up with aquaculture. It's here to stay, and there are challenges in dealing with aquaculture where they want to be in the same places as underwater grasses. We also we're trying to highlight what's been happening with management. Back in the old days, there was nothing. No one really cared. People were more concerned about getting rid of it if it was in front of their house. But again, with the institution of the Bay program, a number of important policies and management considerations were taken. So you can see in the left graph, over time, the number of management policies that were approved by the Executive Council for the Chesapeake Bay. We also wanted to highlight the fact that when I first started my research, you can count the number of papers published in the Chesapeake Bay on underwater grasses in one hand, almost none. And today, if you follow that graph, the top line, the number of papers published on all different aspects of underwater grass in the Chesapeake Bay has been phenomenal, which again shows the interest that has happened in the Chesapeake Bay by our colleagues, students throughout the Chesapeake Bay. And with that, we're going to move into a kind of the case study because we felt it was important to elaborate on that, and we're going to bring in now Cassie and John of the young generation that will talk about the case studies and the penultimate paper that came from our workshop that everyone has been following very closely, including the press. John and Cassie, it's your turn. Don't fall off the stage.
Okay. Well, thank you, Bill and JJ, and thank you for the introduction, Larry, and thank you all for being here this morning and having us to speak. So as JJ mentioned, we're going to walk through a couple of case studies that we did, some analyses focusing on particular species or areas of the bay, to give you a sense of the extremes of trajectories that we're seeing. So I'm going to start with some of the bad news, but then we're going to leave you with some of the good news. And then, as JJ mentioned, wrap it up with the large synthesis of what's going on with SAB in Chesapeake Bay. So the bad news first. This is a paper that we published last year in the Journal of Global Change Biology, and in this paper we're focusing on one species of SAB in Chesapeake Bay, which is eelgrass, or Zostra marina. And we were interested in two things. One, what are the trajectories of eelgrass here in the lower bay? And two, what are the drivers of these trends? So what are the agents that are responsible for some of the patterns that we're seeing? And as JJ mentioned earlier in the talk, Tropical Storm Agnes had a huge impact on the bay. At one point in history, eelgrass extended almost twice its current distribution, shown here in the dark green areas. This is where it was pre-Agnes, and it's contracted into this smaller area, principally in the polyhaline regions of the bay, in its current distribution. And so we're just looking at here in its current distribution from about the mid-80s to present, what's going on with eelgrass. And so in the early to late 80s, we actually started to see an upward tick in eelgrass, representing recovery post-Agnes. It's okay, she can see a lot better now. Sorry. Up until the mid-90s, in which we started to actually see some fairly, I'll just point the things here. We started to see some fairly precipitous declines in eelgrass that have continued to the present day, punctuated by those extreme declines that you can see in the mid-2000s, and then again around 2010. And so since that peak in 1991, eelgrass has declined about a third in the lower bay. And concurrent with this, we actually start to see a shallowing of it as well. Since that mid-80s time period, it's actually decreased its average depth by about 1/4 of a meter. And since pictures speak louder than words, here's an example. These are some grass beds, eelgrass beds off the coast and flats in 1990, and here they are in 2010. And so you can see that all of the grass that was off in the deep areas is now gone, so that cover's declined, and its depth is also declined, so it's now in shallow areas that are closer to shore. So we've seen these trends, both changes in cover and the depth distribution, and we thought, okay, what about our second objective here, which is why? And so to answer this question, we actually drew on some of the survey data that JJ mentioned earlier that's been running concurrently with the SAB monitoring effort. And we used some nonlinear generalized additive models to try to predict cover based on a suite of environmental variables. What we ended up finding was that there were two interesting drivers. The first is unsurprisingly water clarity. As Bill mentioned earlier in this talk, these plants require really high light requirements, so they need a lot of light in order to photosynthesize and carry out their metabolic processes. And so if you start to take light away, i.e. you increase turbidity, you actually start to see huge declines in eelgrass cover. And we believe that this is the primary agent that's responsible for the shallowing of eelgrass, right? As the waters become cloudier, light penetrates less deeply, ergo the eelgrass needs to retreat into these shallow areas, again, in order to optimize its light exposure. The second factor we saw driving this was actually temperature, bringing in the global change angle of this. So what we showed is that up until about 25 or 26 degrees centigrade, there's really no change in eelgrass cover, but after that we actually start to see a decline. I think what's actually really interesting about these data is that this threshold has been identified on a lot of different studies, buckets, in laboratories, all over the world, and we've actually managed to recover it from large-scale observational data, which are very messy sometimes. And so this just goes to show that these efforts can generate and confirm and generalize some of the physiological aspects of these plants. And so like I said, the eelgrass tend to decline with warming, and so we put this together, what we end up seeing is a more complete picture where some of the declines from the 90s were likely driven by turbidity, and then we know that these punctuated declines in 2005 and 2010 were extremely warm events, and so that's where you get this temperature signal coming through. And finally, we showed it's not just temperature alone or turbidity alone that's affecting eelgrass, actually the interactive effect of these two things that's driving its decline. In other words, the water's already pretty turbid, then increasing the temperature is going to have a more drastic effect than if the water was clear. And so we generated this cool interaction surface here, and all this says is, sorry Cassie, if you're at any particular point here along the surface and you give, you nudge in one direction or another, for instance, say you nudge the temperature up, being at a low Secchi depth, you're going to follow the curvature of this surface, wherein you're going to see quite strong declines in eelgrass due to the interaction between these two factors. And finally, I think we don't often stop to ask ourselves, well, why do we care? I think we all care because we love SAV, but you know, I asked my mother, why is this important? And to make my mother happy, we did an exercise here where we started to collate some of the services associated with these eelgrass beds, ranging from nutrient cycling, so things like sediment carbon stocks, nitrogen fixation, to aspects like secondary production. And then, after having quantified those in terms of the amount of area that was lost from 1991, or the most extreme losses, which were noted in 2005, we then tried to ascribe some sort of economic value to the amount of services that were associated with those two quantities. And what we ended up finding is that between $1 to $2.5 billion worth of the Chesapeake Bay economy was potentially lost with the changes in the eelgrass abundance that we've seen over the last 2 or 3 decades. So not only is this an important ecological system, but it's also an economic drive, as a driver of the economy of the Bay. And so this is an aspect that we wanted to emphasize through this work. So that's the bad news. I'm going to turn it over to Cassie now for some of the good news, and then, like I said, we're going to wrap it up with the sort of overarching synthesis. Take it away.
Okay, so we're going to move up the bay, all the way to the head of the bay, in a region called Susquehanna Flats. I'm going to talk about a case study that was part of my dissertation work. So Susquehanna Flats, it's all the way, you're in the head of the bay, in the tidal fresh region, and it's right where the Susquehanna River, which is here, empties into the bay. And all this dark stuff is SAV. So this is about 25 square kilometers, that's a ton of SAV. It's the largest continuous SAV bed in the entire Chesapeake Bay. And it's also really shallow. That's why it's called flats. This is Jeremy Testa and Michael Kemp actually dragging the boat when we were doing some field work because it was too shallow to keep a motor. And so this is what it looks like when you're right in the middle of it. So it's really just SAB as far as the eye can see. And I should mention these are all freshwater SAB species. So a mix of Vallisneria, so that's wild celery, water stargrass. There's some hydrilla up there. And then there's also some Eurasian water milfoil, and then a bunch of other species that are less dominant. And then this is what it looks like when you're underwater. So really, really clear and really, really dense, lush SAB cover. But it didn't always look like that. So this is a time series of SAB, and the shades of green represent SAB density at Susquehanna Flats. So from about the mid-'80s till the early 2000s, it was pretty sparsely populated or sparsely vegetated, so not much growing. And then in the early 2000s, it kind of started to fill in and get more dense, and then it really suddenly expanded pretty much to its maximum extent, so to its kind of maximum depth limits by 2008. And so around here, there was about 50 square kilometers of SAB. Then in 2011, major flood associated with the remnants of Tropical Storm Lee, and the SAB bed declined by about 50%. But since then, it's been pretty steadily recovering. So what I'm going to focus on here is a lot of the synthesis efforts and other sort of large-scale analyses have focused on how external drivers affect patterns and trends that we see in SAB abundance. This work focuses on biophysical interactions, and Larry was a part of this. So how do internal processes, internal interactions between the plants and their environment also potentially affect ecosystem dynamics? And so the internal interactions that I'm specifically talking about are positive feedbacks, where the plants improve their own growing conditions by making the water more clear in their ambient environment. And so we're going to show that these feedback processes likely enhance the recovery or sped up the recovery of this SAB bed, and also increased its capacity to withstand disturbances like storm events. So in addition to Larry, this was also done with Michael Kemp. He was my dissertation advisor. Jeff Cornwell, Cindy Kalinkas, Mike Owens, and Debbie Kinkle. Okay, so one thing we noticed in the VIMS, the raw aerial photographs, was this dark area kind of coming out of the southern end of the flats, especially during ebb tides. And so we were wondering, well, what is that? It looked like clear water coming out of the SAB bed. So normally when we talk about feedbacks, we think about, the SAB bed kind of clearing the ambient environment, their direct environment. And so that can accelerate the rate of growth within an SAB bed. But here it looks like, not only is that happening, but that water is actually being transported to adjacent regions that are unvegetated. And so when you get clear water in these, which happen to be deeper areas, that could potentially lift that light limitation that would normally be there and help the SAB bed expand into those deeper waters. So we did some sampling to kind of test that. So we went out and we measured a bunch of different water quality variables. One of which was a vertical light profile. So we got we measured KD from that vertical light profile, going from the inside center of the bed, this inner edge, through that clear water, we call it a plume, and then down bay from that in these more turbid, deeper waters. And we used that KD estimate together with the depths at in each of these regions to calculate the percent of surface light reaching the bottom. And so 13% is generally what we think of as the minimum light requirement for SAB survival in tidal fresh regions. And that number was derived actually through the first technical synthesis this effort that Bill mentioned earlier. And so in these three regions, so in bed and then in this Clearwater plume region, the plants are getting plenty of light, so generally not light limited. But in this down bay region, so down here, they are light limited, so it's pretty much less than 13%. But if I take the KD data, so the water clarity data from this plume region, and then the depth data from this down bay region to calculate percent light at bottom, it's pretty much almost always above 13%. So there is enough light for the plants to survive. So what we kind of presume from this is that as the bed expands, so as it kind of encroaches into these deeper regions, that Clearwater plume is also kind of gradually creep outwards. And so when that clear water essentially strains out of the SAV bed during ebb tides, it's basically, the bed is increasing its maximum depth limit. And so then it's no longer light limited in these regions. And I should say that when it was at its maximum extent in 2010, the grass coverage went way beyond here. So we know that grass had lived here previously. Okay, so the takeaway from that is feedbacks are also important. So once an SAV bed gets established, and oftentimes that establishment is tightly linked with external drivers like, nutrient loading, sediment loading, and associated changes in water clarity. They can essentially accelerate their own growth, both by increasing internal growth rates within an SAV bed, but also by helping themselves kind of expand into these adjacent, deeper regions. Okay, so I'm also going to touch on what we learned from this major flood event in 2011. So everybody's probably seen this satellite image. So during that time, it was, I think, the third highest flow rate measured on the Susquehanna River. And so there was a lot of sediment loading. So we see this huge sediment plume extending more than halfway down the bay. A bunch of floodgates open on the dam, so high flow rates as well. And so, Susquehanna Flats is up here in direct line of all of that. And so, just like JJ said, I thought that my dissertation was also screwed when that happened, but it wasn't. So some of the grass actually remained, and we were kind of able to use it as a serendipitous, event to learn something. So, in particular, our focus was feedbacks, like biophysical, internal interactions. And so, a really cool thing was that the DNR ConMod program had a continuous monitoring sensor right in the middle of the SAV bed, and also adjacent to the SAV bed at Havergrace. And so that inside-outside bed comparison of continuous monitoring data that started well before Lee and then extended a few years beyond that really allowed us to compare how those differences in turbidity could indicate the role of feedback processes in this bed being able to withstand the storm event. Okay, so we'll start with that turbidity data. And this is plotted as the monthly mean difference in turbidity between the sensor located at Havergrace and the one inside the SAV bed. So the blue bars that are negative indicate that turbidity is higher outside the bed at Havergrace. And then the green positive lines indicate that turbidity is lower inside the bed. Yeah, so higher outside the bed, and then this is higher inside the bed, sorry. And then the bottom graph is the maximum river discharge rate for each of those months. And this big one is Lee, that major flood event in September 2011. Okay, so before Lee, turbidity was pretty much always greater at Havergrace. So clear water inside the bed, more turbid water outside the bed. And that's pretty consistent with what we know about feedback processes. So SAV, slow moving water, they damp the waves. There's less sediment resuspension, and particles also just fall out of suspension, so you get clear water within SAV beds. But then after that flood event, turbidity was significantly higher inside the SAB bed. And so that's not really consistent with what we think about how SAB beds interact with their environment. So we were wondering, all right, well, what's going on with this? And so we analyzed a few additional variables to try to make some inferences about the mechanisms causing that anomaly in turbidity. Okay, so here again we have turbidity. Now this is hourly mean time series. The green is within the SAB bed and the blue is outside the bed. We also have river discharge data, and this is averaged over, I think, a week. And so river discharge is tightly correlated with sediment loading, which is also related to turbidity, right? So river discharge can play a big role in changes in turbidity that we see. I also have wind speed here measured at the Seabeds buoy at the mouth of the Susquehanna River, because wind can resuspend sediments, which can also cause large changes in turbidity. So we want to look at all of these together. I'm going to point out a couple of interesting events here. So this major increase in wind is actually Hurricane Irene. So that happened right before the flood associated with Lee. And during that time, there wasn't much rain, so not a lot of river flow, and just a kind of like a minor blip in turbidity. Then we have Lee, so huge flood event, like very, very high increases in turbidity, both within and outside the bed. And then here we have kind of just a moderate wind event and an increase in turbidity just inside the bed, not outside the SAB bed. So if you compare these two, before the flood event, huge wind event and not a big increase in turbidity, but here just kind of a moderate wind event and a big increase in turbidity just inside the SAB bed. What we infer from this is that the flood deposited a lot of sediment on the flats. And now all that loose, unconsolidated sediment is being resuspended by the wind and causing this really huge increase in turbidity just within the SAB bed. And I want to point out that that pattern was pretty persistent through 2012, so for the year after that flood event, and also 2013. And I think I kind of stopped this analysis around that time, but I think this pattern kind of continues into 2014 as well. So it's interesting that there is this pretty long-term memory of that flood event in this system. But the thing that's even more interesting to me is that in the summertime, the turbidity pattern kind of goes back to normal, where it's lower inside the bed, higher at Havre Grace. So there's more clear water within the SAB bed. And so that's suggesting that as long as the plants can kind of push through that turbid water in the spring and form a nice dense summertime canopy, they can clear that water up by preventing that resuspension by the wind. And I just have this up, I'm not going to explain this in depth, but I have this up here just to note that inference that we drew just by looking at those data was quantitatively robust. So I ran a mixed effects model where I looked at how the effect of wind on turbidity changed both after the flood and by season and across space. So it wasn't just that it was less windy in the summertime, so you didn't see that resuspension effect. It was the actual effect of wind was greater after the flood, or what did I just say? I think I said that backwards. The effect of wind on turbidity was greater after the flood event, and also it was greater in the spring compared to the summer. So again, there was just less wind-driven resuspension in the summer, probably because the plants were there to prevent it. And so, this all supports the idea that that these feedbacks can enhance the capacity of these SAB systems to withstand major disturbances. So again, feedbacks are important. If an SAB bed is big enough and it's dense enough, it can essentially protect itself from these types of external stressors. And also, we can kind of extrapolate these dynamics to other places. But Susquehanna Flats is also very different from a lot of places. But the case study, this kind of like in-depth case study approach, does a good job of really helping us understand the mechanisms by which these processes are happening, and particularly the effect of these really local scale processes on the dynamics of the system in general. And so I'll wrap it up by saying that now what I'm interested in investigating is now that these SAB populations are recovering, so not just Susquehanna Flats, but most of the tidal fresh and lick-healing regions throughout the bay, which is kind of a teaser to what John's going to talk about. So now that they're recovering, how are they affecting broader estuarine ecosystem processes? So one, to what extent is all this additional plant biomass on the bottom affecting the metabolic balance of the ecosystem? And then also, they can, not only are they trapping sediments, but they're trapping the particulate nutrients attached to those sediments. And they also can assimilate a lot of nutrients. So how are they affecting the nutrient dynamics of the system, both now at their current extent, and also, if they continue to recover and reach their maximum extent. Okay, and so now I'm going to hand it over to John, who's going to talk about the drivers of these large-scale recoveries throughout the Chesapeake Bay.
Right, thank you, Cassie. Try not to blind you during this section. Right, this is the coup de grace, right? This is bringing it all together. We've seen some perhaps negative trajectories with eelgrass, these positive feedbacks that are sustaining large cover in the Susquehanna Flats. But what's going on in the entire Chesapeake Bay? And so I'll point out that this question's already been answered by us in a paper that came out earlier this year in the Proceedings of the National Academies of Sciences, suggesting that changes in the nutrient regime here in the Chesapeake have actually led to some good news. And I just want to remind you again of the programs that JJ mentioned at the beginning of the talk, these large-scale bay-wide monitoring efforts, both the aerial photography that's measuring cover, as well as these associated efforts that are both observing environmental variables and modeling environmental variables. And so what our ultimate goal of this synthesis group was to bring all of these pieces of information together in a single unified framework to be able to answer those two outstanding questions that Bill mentioned at the beginning of the talk. What's the trends of SAB in Chesapeake Bay, and how are human activities implicated in those trends? And to answer this question, we used a technique known as structural equation modeling. So this does what I just described. It takes a whole bunch of variables and unites them together in a single network. I'll make a note that these are all observed variables, so they're things that are measured. There's one instance where we included a model variable, but that distinguishes it from some other efforts that you may have heard about at the conference. The other defining feature of structural equation models is this term causal. So why don't you raise your hand if you've ever heard the term correlation does not imply causation. Okay, how many of you believe that? Okay, that's not surprising. What I'm here to tell you today, though, is actually that statement is not correct. Correlation does imply causation. The only problem is we don't know the direction. We don't know if A causes B, B causes A, or both of them are being driven by some other unmeasured factor. However, we have lots of evidence to draw on to be able to define the causal space and constrict those potential directionalities. So for instance, SAB are not spitting rays of sunshine that are shooting back into the sun. We know that there's an inherent physical directionality. We also know through experimentation and prior correlative analyses that things like nutrients are perhaps central to this story. And so we can craft a model that we can ultimately start to draw some causal inference. And because we're uniting all of these variables together in a single framework, the structural equation models are ideal for testing these cascading or indirect effects, as you'll see in just a moment. And it's actually just traditional linear regression under the hood. So if you've ever done a regression model, you've done SEM, just a very simple version. And very quickly, the notation we use is that we have two variables here, x and y. We say that they're causally or directionally related here with an arrow going from x to y. So we would interpret this as x causes y, y causes z, this network of equations is our structured equation model. Okay, so in order to answer the overarching questions of our synthesis group, we took two separate approaches. The first was focused on about 100 independent sub-estuary watersheds. In this case, we wanted to understand how human activities in these watersheds, things like farming and urbanization, led to downstream effects, those cascading effects that I mentioned, that ultimately influence the amount of SAB that we find. We paired that with a second analysis that we then extrapolated to the entirety of the bay, looking at what happens when the nutrients that are generated by the activities that are coming off these watersheds actually hit the water column, and what are the mechanisms that are operating there to change SAB. So a large part of our goal in these workshops was to actually sit down and hammer out a causal model that had strong support, that made sense, and that reflected the data that we had available. And so this was the model that we managed to converge on for our first analysis of these sub-estuaries. I will walk you through it. It's actually a lot simpler than it looks, mercifully. So the first relationship we uncovered is that as you increase the amount of land that's devoted to agriculture, you increase the amount of fertilizer that's supplied. That should be no surprise, and thank God we recovered it, because if we didn't, then we'd have to question everything that came. The second relationship we see is that as fertilizer application increases in these watersheds, we see an increase in non-point source nutrient loads. So these are from the Chesapeake Bay watershed model. And we found out that these loads actually interact with flow. So this is rainfall in the watershed that's coming off the watershed. It's the delivery mechanism for these non-point source nutrients. So these two things interact to reduce SAB. In other words, the more nutrients you have in a watershed, and the greater the flow, the greater the delivery, the less SAB you're going to end up with. The other interesting thing we found was actually a positive and independent role for biodiversity. So in this case, we had the number of species in each of these sub-estuarine systems, and the more species that were present, the more cover we had, which was sort of unexpected, but actually is starting to emerge as a fairly general pattern in many other systems, and even in some seagrass systems like the Indo-West Pacific. And I'll note that we ran this model separately for each of the salinity regimes in the bay, because we know they're very different species compositions. But across all those models, which showed us virtually the same things, we had an explanatory power, an R squared, ranging from 0.67 to 0.8, which was quite good, we think, for a very complex system. So I mentioned a second analysis, what happens when these loads hit the water. And so this we're drawing on a bay-wide analysis in monitoring data that's collected both in the sub-estuaries and in the main stem. And so here what we saw was that nutrients increase chlorophyll concentrations, so they stimulate phytoplankton. That chlorophyll clouds the water column, so it decreases the clarity. And as we've established already, clarity is good for underwater grasses. They like light. And so this actually aligns with some of these conceptual diagrams that were generated as part of the early technical syntheses. What we've done is actually taken data and validated them empirically, which I think is a really exciting application. And so what we end up here is a cascade by which nutrients increase the turbidity of the water column, and that turbidity decreases SAB. And like in the sub-estuary analysis, now looking at the entirety of the bay, we again find a role for species richness. So as you increase the number of species that you find in a particular area, you end up having more SAB cover while controlling for instance, the influence of nutrients. So what does this all mean for the bay? Well, good news. Nutrient concentrations in the Chesapeake have declined on average by about 25% since the mid-1980s. And consequently, during that same time period, we've seen over a 200% increase in the total aerial cover of SAB in Chesapeake. So this is a good news story in the sense that management efforts that have been instituted to limit the amount of nutrient loads that are changing properties of the water column have generated measurable impacts and increased the density of SAB in Chesapeake Bay. So we've done it. We still have lots more to go. As JJ mentioned in the bioscience paper, there are some management goals that we've set for ourselves. We're only about 50% of the way there, but these data suggest we are exactly on track. So continued measures to limit nutrient inputs, as well as perhaps new measures to, for instance, conserve biodiversity, may continue this positive trajectory of recovery. So if we return to these original two questions, what are the long-term trends of SAB in Chesapeake Bay? Well, despite some variation, for instance, eelgrass versus the flats, on average, we see that SAB is increasing in Chesapeake Bay, and that is potentially a direct consequence of our ability to limit nutrient inputs during this time. So we're kind of close on time, but we're going to switch it out, and JJ and Bill are going to wrap it up.
Okay, the story doesn't end there because in 2017, in our annual monitoring, we saw some very interesting results occurring off Solomon's Island, which hadn't had SAB for 40 plus years. So the picture you see there was from our aerial imagery in 2017. I remember seeing this and calling Jeremy and said, Jeremy, you need to get out there and see what's going on. And sure enough, SAB has returned to Solomon's Island for the first time in 40 plus years. So those little dots you see there are patches of widgetgrass. And I asked Jeremy a few weeks ago, would you go out and check it? And those patches are still there. So the recovery process continues. We're really excited. And so it's pretty impressive. Of course, this year, we all know this is going to be a test year given the amount of water that we've had in the bay. So the resilience issue is on the table right now for what happens in 18. And just to quickly, we've been presenting these results at a number of scientific meetings at CERF. We want to let our colleagues know. We've been getting very positive feedback from our people all over the world about just how important the study was.
We also decided that this story needed to be told beyond our peer science group. We wanted a bigger media audience. So we actually contracted a guy called Mike Smith and went to the National Press Club in Washington, a little message triangle. We got our story together and we pushed this out. It received a lot of media attention. It got Washington Post 2 editorials, which actually went beyond the documentation of the SAB resurgence and said, maintain the funding for Chesapeake Bay program. JJ and I wrote an article for The Conversation, which is an online kind of blog, which got picked up word for word in the LA Times and Houston Chronicle, etc. All in all, about 56 million people were exposed to the story in traditional social media, well beyond the 20 million of the watershed.
Okay, so the story again doesn't end there because remember what I said about Rich? Rich wanted everything since biblical times, about everything that we ever knew about SAB. So we had to get back to, well, what was going on in individual segments. We knew the big picture, but we also wanted to provide relevant information for the managers. They're the ones that have to deal with the watershed implementation plans and all the other things each community has. So we stepped back and said, okay, let's look at what's going on within the individual segments. We met and set up a segment matrix analysis. We set up 5 key questions about what was happening historically, whether the segment was meeting its restoration goals, some of the key historical issues that are going on within each segment, the vulnerability and resilience in each of those segments, and then a comment on what are the management implications for each of those segments.
So I'm going to, these fact sheets we generate for each of the resource managers have specific data and recommendations for each region, time course and conceptual diagrams for each region, some take-home points, short summaries linked to the data, and an overall stoplight color scheme. So, dark green is attained, we've attained this, and it's definitely doable, all the way back to red, likely unattainable.
Okay, so we knew there were a lot of segments. Some of these segments we combined because they were linked very closely by just proximity. And this is that segment scheme from the Bay. And I'll run through 3 examples since we have to have 57 of these fact sheets. These are 2-page fact sheets. They're real simple. They're data rich. We knew that managers have access to a lot of data, but we wanted to provide very relevant information to managers and very simple diagrams that they could then use from that point on. So this is the Susquehanna Flats example. 2 key figures. You've seen the top figure in Susquehanna Flats. We adopted Cassie's technique of using the flow of SAB coverage over time from 1 to 100%. We also developed these interesting figures that use icons of what was actually happening and the processes influencing SAB and how it changes the abundance of SAB. So you can see icons specific for species that are in that region, and the density coverage represents the coverage that you see, for example, in flats based on how the event was developing, and the time course of events for the flats in terms of turbidity, and also you can see where Irene and Lee popped up, and the development of the processes that contributed to its decline, and then also an issue that we're dealing with fleeing beasts. Every one of these fact sheets has these two diagrams, and then the stoplight features, and then the second page of this fact sheet for these managers, we go into a one-paragraph description of, again, whether it's met its restoration target, or if not, what it's going to take. We have some information on the historical coverage, again, some of the vulnerability resilience issues. So one paragraph each, real simple, but again, supported by quite a bit of data in the numerous reports that many of you have. This was the Object Bay. So you can see the previous one was green. They had met its restoration target. This one is the Object Bay, which is well below the restoration target. And we said for it to meet its restoration target, it's going to require quite a bit of improvements in water quality. Another interesting icon that you see, some of these places we've attempted restoration via transplant, so we have a diagram there. But every one of these segments has each one of these elements. And in this particular case, Lindhaven, there's the next worst one in orange, where to get SAB back to the Lindhaven, it's going to take a lot of hard work to really improve water quality to get SAB back, because it's a decline to where there's really no more SAB in the Lindhaven.
So kind of reflecting now on this, we call it our SAVSIN process. We have to have the enabling conditions. We have to have a compelling topic. We have to have resource managers. We have to make them happy. We had to have nominal support. Not a lot of money, but just enough money for travel and accommodation. And you had to have some gray beards in there to sort of set the scene and have experienced leadership, and then recruit the young, smart people, but not that many. A dozen max, and that's the NC's model as well. You don't do a typical stack workshop 40 to 50. You don't get anything done in this context. We did multiple immersive workshops for them, 3 in our Annapolis office, 1 at Chesapeake Biological Lab, and then the products that come staggered after the series of immersive workshops. We had a lot of communication. This is 1 day of emails from JJ. I'm not kidding. And so lots of communication. We had to be kind of flexible. Our agendas were very crude and simple. We kind of self-organized. We also used some of our funding to bring a researcher, Chris Patrick, up from Texas A&M. He had been at CERC and had a good history and knowledge, and is a critical member of our team. We also focused on products, the peer-reviewed papers you've already heard about, the segment analysis we just heard about. I think it's important to have a conducive location. You have to have places, giant whiteboards, and as you see in our Annapolis office, but places where we walk to the restaurants, coffee shops, bars, and hotels. We didn't need to get into cars and navigate. Even in the Rand's Head in the bottom right, you can see we wrote on the napkins, the beer coasters, our ideas of what SAB meant to us, made a little work plan. We have to have clear goals and objectives, as we've discussed. Ultimately, to make this something people want to do, we also have to have fun. So this is our bid emoji of JJ. This is our logo on a coffee cup, our poem, the Chesapeake Sentinels. In the future, one of the things we didn't do at the beginning, but we did toward the end, is we brought in one of the critical staffers from the Bay program, Melissa Merrick here, who did all the graphics that we've been celebrating here. And so we should have had her from the beginning. We should have maybe brought some grad students in. Brooke Landry from PNR was a critical member throughout the team, the segment analysis as well. JJ, of course, with the beer in the background. I'm gonna end with a song, because when we went to the National Press Club, one of the reporters, the Greenwire reporter, said, "So what's the meaning of this anywhere else? I mean, is it really if you can make it here, you can make it anywhere?" And you come up with that little line, and you give me some beer, and a dangerous combination occurs, and that inspired a song. It's called Chesapeake, Chesapeake. And The voice you're going to hear is Melanie Jackson, who's Jeff Cornwell's graduate student at Horn Point. And last Christmas, at the Christmas party, she did a little a cappella rendition of Let It Go, and it was like, we got talent here. My daughters say, Daddy, stop singing. You have singing issues. So we got talent. And we have a video team now, Jamie Curry, but also Ann Swanson's son, Sky, helped out putting this little video together. So, are you ready for this?
Yeah. As long as you're not singing, Bill.
Can you hear it?
It's not where
It's better.
I want to be a part of it. Chesapeake, Chesapeake. They are resounding with praise, and they are
It's buffering.
the heart of it. Chesapeake Bay, Chesapeake Bay.
Is this on the...
It's the message.
I wanna wake up on a bay with grasses grow.
let's see, I'm going to do small, so video.
And towards your big and bare top of the heap, your Chesapeake, they're melting. Let's make a brand new start of it in Chesapeake Bay. If you can make it there, you can make it anywhere. It's up to you, Chesapeake,
What a show. I mean, this was just great. This is, coordinated and choreographed, and it was just wonderful. Thank you so much, all four of you. I'm sure there are lots of questions. Are there any immediate questions that I can get from the audience? Yeah, go ahead.
That was a terrific set of presentations. I have one question, which is, do we know how the phytoplankton populations are responding to the expansion of submersible grass?
Good question. Are the phytoplankton responding to the SAB? Probably not on the Bay-wide scale, no.
Well, but I meant locally.
At the local scale, yeah. I don't know, Cassie, did you measure chlorophyll?
Yeah, I mean, we use the chlorophyll monitoring data that was measured in the flats and adjacent to the flats, and it's definitely lower locally within the SAB beds. I mean, part of what my research now is trying to look at is, are they retaining enough nutrients to limit bay-wide phytoplankton production? Or if they're not now, like how much more SAB do we need to get to that point?
One of the things Cassie showed in one of her papers that she didn't show here is that nutrient levels inside the bed are extremely low. I mean, much lower than they are outside.
That is actually a very interesting question, and we didn't look at the large-scale trends in chlorophyll A, but I will say this. There were some interesting relationships that I didn't get into that suggested that there were actually direct effects of nutrients on the SAB itself, which we interpreted as overgrowth by epiphytes. And that was actually twice as strong of an effect as what we saw from the water column, chlorophyll, and then the changes in turbidity. So it's likely a combination of both things that are occurring, but we're actually missing that critical epiphyte data that could help shed some relationship on those two mechanisms.
The image that Cassie showed regarding the flow emanating from these beds that are crystal clear, that's just one example. We see this in our imagery in many areas, in particular in the Washington, D.C. area with the dense hydroma beds. Huge plumes of very clear water emanating from these beds on and at a time. So sort of indirectly getting to your question about chlorophyll inside these beds.
In back, go ahead.
Yeah, my question is, you said you saw an increase of like 215% in seagrass. Can you say how much of that was due to restoration efforts or natural regrowth? And would the trends connecting land use and nutrients have been strong without the restoration?
There has been some restoration efforts, but most of it's been natural spread. We have been tracking areas that we have done specifically targeted restoration, but generally throughout the bay, the major effort has been just natural spread of existing plants or from seed banks that occur within these small marsh creeks.
Go ahead.
I have a question about the survival of Susquehanna flats within the context of sea level rise. You described the process of sediment being deposited and then resuspended and leaving. So there's net transitions with these high flow events. We know that tidal wetlands need sediment deposition to keep pace with sea level rise. So my question is, do you see sediment supply to the flats in that way as important for the survival and even growth of the flats within the context of sea level rise?
Well, I can start answering that one. In the context of the amount of sea level rise that we've seen to date, I'm not sure that it's enough to have a major effect on the light penetration through the water. But so the Susquehanna Flats is a depositional environment, and there are people in this room who are studying specifically deposition over time. So I don't know, Larry, do you want to?
To clarify, I think my question is, you need to maintain a certain
Right.
depth.
So let me address that a little bit. All sediment is not equal, right? The Susquehanna Flats is built on a sand base that was built before the dam was put there. The dam has effectively cut off the sand at this point. So there's a little bit of evidence that the shape, the morphology may be not growing as fast as it used to. The fine sediment that Cassie's talking about that causes the turbidity in the water column is a surface phenomenon. It's not really related to the growth of the bed. It's related to a fine film that's near the surface or on the surface, and is deposited temporarily and then washed away.
Okay, I get that to belabor the point, but I guess then my question is deposition on this deposition feature important for the future maintenance of the flats as an entity that we love?
Yeah, I think if we look at long term, that there are grass beds now in places that were salt marshes. The salt marshes.
Right.
And we know from salt marsh studies that you need an accretion due to sediment-trapping organic peat development to be able to keep up with sea level rise, for them to maintain sea level rise. So presumably the same thing. Seagrasses, at least in Chesapeake Bay, do not produce peat. So the only way they can do accrete is through sediment deposition and or migration shoreward.
Go ahead.
Cassie, you mentioned hydrilla. I know in the early 1990s, everybody was up in arms about it. The head of DNR had declared war on it. I have a couple questions. What's the current status of hydrilla today, and have we defeated it? And then my second question is it good for anything, or is it just in total competition with our native grasses, and so we just hate it?
Well, I can only speak about Susquehanna Flats specifically, but there are a lot of native species there as well. So I mean, just off the top of my head, I would say hydrilla is maybe 10-15% of all the species there. So there's also wild celery and water stargrass. I would say they make up the bulk of the distribution of SAB on the flat. So also hydrilla, just in terms of the biophysical interactions, forms these really dense canopies. So it does a really good job of helping to clear out the water. So if you're, it just depends on what your goals are. So if your goal is to create an environment that's conducive to SAB growth, whether it be natives or non-natives, hydrilla does a really good job of that.
I'll add to that because I've been following the hydrilla story from day one when it was first accidentally introduced into the Potomac River in the early 1980s. Someone thought they were planting Elodea. It turned out to be hydrilla. And so we've been following the story, in particular the very interesting interactions that occurred early on when people wanted to get rid of every spring and they knew they couldn't. Well, the rest is history, and hydrilla really is in every estuary in the Chesapeake Bay. What we saw in the Potomac River was a very interesting phenomenon. While it did spread, we also began to see the recovery of many of the natural species of SAV that weren't there prior to 1983. As a matter of fact, the Potomac was devoid of any vegetation from the early 1900s due to the mess going on in DC. And this was all documented by Nancy Redvicki and Virginia Carter at the USGS. So we were really interested, and Nancy's provided some really neat data that we have in our website of all the species that now occur. Many of these are natives that occur in Washington, DC. And there was an interesting controversy surrounding Dyke Marsh recently because people thought it was just hydrilla that was occupying all these areas because they wanted to rebuild Dyke Marsh. The fact is that these beds are really rich in numerous fish species, and in some places, for example, the Pamunkey River or the Mattaponi down in the lower New York, lower bay, it's the only species, and yet it provides many of these deemed ecosystem services and systems that have not had it. And of course, as we said, as in the Potomac, and we saw at the flats, it's become just a natural part of the community.
Go ahead, Don.
Yeah, JJ, by the time you hang it up and go home and grow kale, you're going to be at this about 50 years. Fast forward another 50 years. You've got the TMDL is mission accomplished. You have half a meter of sea level rise, an increase of about over 1.5 degrees Celsius temperature, and a rapidly retreating shoreline as the bay not only gets deeper but broadens. What's SAB going to look like?
Well, it's possible that we may not see as much velocity today as we have because it's going to be primarily in areas closer to the mouth of the bay, mainly in the coastal bays. We may see a substitute for some species that are in North Carolina, like the Dennison today, for example, what's called shoulder grass or phylogeny. I think the freshwater species are going to do well. I think they're going to be okay because they're going to be responding to a number of different factors, primarily salinity. So, like for example, this year, as a prelude to the excessive amount of fresh water, we may see even more freshwater grasses because the salinity is now depressed. Because that is a key element. These are grasses that don't like salt. So in the future, yeah, and then there's widget grass that we have seen in the last couple of years just dominating the lower Bay area. So I can't really forecast because I don't want to get into forecasting, but I'd say we're going to have SAV, but they're going to be probably very different communities.
One other thing you did mention that's happening is acidification. And when corals disappear, what wins out are seagrass. Seagrass like high acidity because they need carbon dioxide. And it turns out when you go to natural CO2 seeps like off New Guinea, the corals are pretty much gone and the seagrasses are thriving.
So it'll be a nuisance.
Hopefully.
Bill?
I'm going to follow Scott Phillips' earlier advice about trying to come up a little closer. And I want to take the prerogative of a co-organizer. So we're about 5 minutes from break. And Harry, I know you have a question I want to get to. But I just want to take a moment, first of all, to thank JJ and Bill and this entire team for what I think was a fabulous presentation. You guys really obviously put some time into it. It was a wonderful synthesis project that really, the fourth of what I understand is a synthesis project. And it was nice to see a collaboration. It involved VIMS, UMCs, people from CERC. I saw Don Weller up there, Patrick, we mentioned, and Chris Patrick. And I saw Rebecca's face and Nathan there, and she's a Hopkins representative. So, or at least to me she is, because she worked with me. So it was just a great presentation and a great synthesis study, and I want to really thank you guys for putting that together. And you mentioned Rich, and we've had one ovation for Rich. Rich put together this synthesis. Some of the earlier, if I understood correctly, it was at least 2, maybe 3 out of the 4 synthesis studies he had a lot to do with. And beyond that, I want everyone to know that he's also been a champion for reinstigating synthesis projects for staff to oversee within the Chesapeake Bay program. At least we have a placeholder. It's not extremely well funded, and we don't have the same kind of wonderful background you do on every topic to do as well. But it's going to move forward, and I think it will help the topics. We talked about that this morning. It'll help a lot of other things. The first one's going to be on climate. And so I think it's time for standing. This is the last chance we had as a group, I think, because it's the last plenary session, to thank Rich, because he is retiring, and I think he deserves a standing ovation. I have 3 other points to make, so forgive me for the time. I'll try to be quick.
One, you can make one.
One? Oh, we've got a little time, don't we? You've got 5 minutes to end. Okay, I'll try to be very quick.
Pushing the lunch a little bit.
Okay. We can push. It's 11:45. Yeah, okay. So just 5 minutes. I want to really mention Mike Kemp. I think his name was mentioned here quite a bit. He's retired. He's no longer here, and obviously he's a faculty member who's retired and had a lot to do with a lot of this. Cassie's advisor, and I think we should really think of him and all the good work he's done. He's down in South Carolina now, hopefully enjoying retirement, but he wasn't able to make it here. So I want to remember him. And the other thing I just want to briefly mention, I'll make it 4. The last thing I want to briefly mention is there was a slide up there of 3 guys standing there in 1972. And so there was one minor correction to what was said, which was that the modeling and monitoring joint effort and research began with the program as a real endeavor in 1984. I'd like to point out that it really began in 72 with that group, and it was a bay model, a physical bay model, not particularly well conceived, but because it was before computers. That happened in the early 70s, and there was a lot of research. There's a great document in our offices. It's called the Chesapeake Bay. It's about this thick, and it was written by those 3 guys, and also involving CERC. Moving forward to develop the science that led to the production of the Bay program. So I just want to make that little plug, if I may, and I'll sit down, Dave.
Yeah, go ahead.
I'm Wei Jun Hai from University of Delaware. So its pH is so high in the SAV bed due to very high biological production. So PCO2 must be very low. I wonder if anyone studied the carbon concentration mechanism. How does these plants get CO2? Just one
That's a really good question. For a long time, we've been talking about boundary layer diffusion limitation of carbon dioxide, particularly in these dense beds where the pH goes up to 9 in the middle of the day. So absolutely, they use a variety of different mechanisms for DIC porters across the cell walls and stuff. So they also do a lot of recycling in their lacunae, in the air canals. So the CO2 concentration in the air canal is about 3.5% instead of 0.3%, order of magnitude more than atmospheric. And so they do a lot of recycling. And so we see that with the Del C13 signal as well. So they're a functional, there's a lot of CO2 conserving mechanisms in plant metabolism, C4 and CAM metabolism. Seagrasses use traditional carbon metabolism, C3, so to speak, but they effectively become like the C4 or CAM plant, but they're recycling with those gas mixes internally.
And I'll just plug Wei Jun's talk. You're going to talk about how Susquehanna Flats affects carbonate chemistry of the bay, right? Is that your talk? So see Wei Jun's talk this afternoon about how the carbonate chemistry of the whole estuary can be affected by SAD metabolism.
Yeah, my student Jim posted here. So when we talk about carbonate precipitation, maybe when you precipitate the carbonate, they also generate the CO2. So could it be locally a source of CO2 for the plants?
So, okay, I think I'm being told that we have to end this. I don't want to. But let's thank our panelists one more time for a great presentation.